Dear students, hope you all are doing well. Welcome to another exciting session of the course Mechanical Characterization and Modeling of Tissues. Right. So today we are going to continue our discussion on clinical testing, which is one of the primary aspects which have to be followed while dealing with any soft tissues from the cadavers, which is a dead human model or an animal model, for example. Like, so all these testings are important to validate all your mechanical model, finite element models, all sorts of experimental soft tissue models which we have been talking about and discussing like through the course, right? So let's get started with the discussion, and today we are going to be talking about some of the very exciting case studies on where actual cadaveric human skin have been tested, and we'll look at some of the opportunities and options there, right? So let's get started. Okay, so we were here in the last lecture. We were talking about some of the different levels of testing, clinical testing, why clinical testing is important, what is a preclinical versus postclinical or a clinical, different stages, right? More of a pre and post market kind of categorization have been followed here. So we're looking at that, and we studied about what's a pilot and what's a pivotal study. Pilot is where you're looking at a smaller population, right? Trying to figure out that what are the safety standards and what are the results with this particular population when we include a tissue construct or a biomaterial or a medical device. And pivotal study is of a known fact where we have a larger population and we are just looking at the efficiency aspect of things from the uh, example is like, for example, a new kind of skin graft if applied to a range of human population after a lot of testing with certain machines and meshing devices, right? And we are looking at the overall outcomes of the same, right? Preclinical typically has in vitro and in vivo studies, which includes animal models, and basically you will see a lot of research publications which are focused on this preclinical studies, and clinical is more of when you're getting into the human body, right? So we're going to continue on this discussion. Let's talk about randomized controlled trials, which is the stage 3 of things. So here you have practical considerations are likely to lead regulators and policymakers to define a limited category of devices for which stage 2 studies and stage 3 randomized controlled trials will be required. This is not required for all sorts of devices, right? The evidence that inadvertent patient harm can result from apparently small modifications to existing devices suggests that a clinical trial should normally be required for most implantable devices. What this means is, uh, even though certain implantable devices in a certain category may be working just fine, but you have developed or innovated some new implantable devices in the same similar lines, but maybe you've changed the material, maybe you've changed certain design parameters, and that could have very chaotic effect on the human body, which you may not be able to predict. That's why any kind of such changes, even though there is a lot of literature supporting the usage and clinical trials of these devices, needs to go through proper clinical trial again. That's the point, right? So anything which goes into the human body, even though let's say for a skin tissue construct may have worked fine, it doesn't mean the liver tissue construct is going to work. Or for that case, like a tissue construct which has worked in the arm, right, like due to a burn injury when it was used to replace the skin there, may not work fine in the facial skin, right? So all these aspects have to be looked together into further clinical trials or randomized controlled trials. Then we have stage 4, which deals with long-term studies, right, where you basically have a post-market surveillance conducted from a number of reasons, like, right, including to confirm the safety and efficacy of the device once it is on the market, and to answer questions about long-term safety or performance of the device, right? So the long-term effects are also very, very important. So there might be this tissue construct or biomaterial or medical device which we have developed, and this has passed through all the clinical trial phases. It's working well with the human body, but this may not work after 10 months, for example. So that's where the long-term studies becomes useful, right? Or a particular implant may fail after 100,000 cycles or something like that, right? So These long-term studies will only be able to declare such results that what can be harmful in the future, right? So prospective registries were started from the outset of clinical use. They could be used continuously for safety surveillance, right, of higher risk devices. So higher risk devices are very closely monitored, and just so that like the risks do not magnify and there is no cutting corners around the manufacturing and implementation of such devices. Picking up weak signals from rare events and outlier device reports as technology proliferates, right? Like, so there would be cases as a new technology kind of grows more and more popular, as there would be rare events when there may be an infection case or a death case, but these have to be considered, in particular with high importance. That such a thing should not be repeated anywhere. If it is a repeat occurrence, then there might be something wrong with that device adaptation or tissue construct adaptation across a wider population, right? Okay, from a ISO 14155 clinical investigation of medical devices for human subjects, norms. Clinical investigations must take into account scientific principles underlying the collection of clinical data, along with accepted ethical standards, which we have talked about, surrounding the use of human subjects, right? So this is very, very important as per the ISO 14155 norms. A properly conducted clinical investigation, including compliance to the clinical investigation plan and local laws and regulations, ensures the protection of human subjects, the integrity of the data, and the data obtained is acceptable for the purpose of demonstrating conformity to essential principles. So whatever set of data which you are getting generated through this clinical trials, they have to conform with these realistic scientific principles. So it should never happen that I develop this new tissue construct for some organ replacement or some tissue replacements, and it is leading to much more failure cases than possibly success cases, or let's say 7 out of the 9 cases are leading to infections, then that is violating the principles, right? And second is like the integrity of the data has to be maintained. It should not be that somebody is manipulating those data. That is from an ethical standpoint. And enough data has to be produced to properly conclude certain things. We should not be testing a new uh, tissue construct or a biomaterial on 5 human subjects and tell, oh, this is highly efficient. It should be tested on at least 500 to 5,000. There are statistical norms into how big a certain population this particular disease is affecting, or particular injury may be affecting, and based on that there are specific statisticians like who will tell you like these are the lowest numbers which you need to conduct such a clinical trial. So that has to be confirmed with, right? From an ethics standpoint, and we have discussed this at length, but just to highlight, it should generate new data and answer specific safety, clinical performance, and effectiveness questions, right? That may remain unanswered by the current body of knowledge, right? So this is the overall goal of from an ethical standpoint. The desire to protect human subjects from unnecessary or inappropriate experimentation must be balanced with the need to protect public health through the use of clinical investigations where they are indicated, right? So any kind of unnecessary or inappropriate experimentation should not be performed, right? So I should not do like I've developed this new biomaterial or medical device or tissue construct, and I want to use it on, let's say, some poor population because they do not understand how this works or how this may affect them, and let me get some data. That's totally ethically wrong. So those kind of things should not be done. It should go through proper channel, and it should have a justified need for why this needs to be tested. Yeah, let's say this has been tested on animal models, several trials have been conducted, and now you feel that this is the next step, natural step, where this needs to be tested on the human body. Yes, but not like you print a bioprinted construct and the next thing what you want is to test it on the humans. That's not the norm, right? So this is how the right ethical practices have to be followed, um, right? So from designing of a clinical trial, there are several things which we should consider, right? First is there should be a clear statement of the objectives. This is followed by minimization of the risk to the subjects, right? This is very, very important. defining the adverse events, right? So what are some of the adverse possibilities or events which may occur, right? Things may go wrong, many things may go haywire, or we may not have conducted a cytotoxicity study, for example. This should be properly stated. First of all, you should conduct the study if something is missing by that table which we discussed in the previous lecture. But if you have missed out on any of the mandatory tests, first is you should not go for a clinical trial, and second is if anything was considered unnecessary and missed out that should have a possibility of like adverse effects, that should be declared. Right? These are some possibilities. You may have a infection due to some kind of injury and other things which you may have been encountering. So let's say this particular type of skin scaffold has been built for burn patients, and you are trying to treat an electrocuted patient who has lost a lot of skin, like in some certain places, and you're trying to use this particular construct for that. So you should clearly indicate that these tissue constructs have been tested for burn patients and for burn possibilities, and we are trying to test it for the very first time on this in this case. Maybe because you did not find a similar setting, or you were not able to create a similar setting. So these kind of things have to be clearly laid out, right? Defining the study outcomes or endpoints, that's also important, that what are the endpoints. Appropriate study population have to be selected. So very often you would observe this like across research studies, that you want to test for a certain disease, tissue related disease, and what you end up doing is testing on 5 or 10 healthy subjects. That's not the way to go, right? You have to get folks from the correct population and test on them in order to derive right results, or possibly have a clear path towards solution of that problem, rather than so publication versus like actual work which can go for clinical trial are two very different things, right? Sampling strategies, the right sample of the population needs to be maintained, right? That's through statistical analysis, measures to minimize the bias. So it should never happen that, okay, I'm going to take these two kinds of population and this is what my expected results are. If I get that expected results after testing on 15 people, I think this is going to be correct. No, that's not the way. That's introducing a bias. If you have a clear bias, then there is no use of doing clinical testing, right? Clinical testing is done to make sure that there are no biases, and it's a very clear set of data which we find out, with outliers and cases which might be faulty. Identifying the confounding factors is very important statistically. Selecting the appropriate controls and comparator devices like active controls, historic controls, and whatnot, right? Study design is important. Is it a parallel study, crossover study, single arm study, or a cohort study? These are different categories of studies, right? I'm not going to get into the details of all these because this is more to do with clinical trial. It's just good to understand these norms, because this will travel around all your efforts to do all sorts of animal and cadaveric testing, right? Type of comparison, you can do a superiority comparison, non-inferiority, then equivalence comparison. Is a particular kind of medical device or tissue construct behaving the same way as how a traditional device should behave, or is it behaving superior to that, inferior to that? So these kind of details can be chalked up, right? Follow up duration and monitoring. So what is the time after which you are going to follow up and look into what is the healing with this particular tissue construct, or this healing patch, wound healing patch, right, for example. So these durations and monitoring time points have to be clearly laid out. So that brings us to the gold standard of study design for clinical trials, is a randomized control trial. Randomized control is where you have random human population without any known demographics, right? And here typically in a drug trial you will have placebo where nothing is provided, versus a surgical intervention, could be a medical device or a tissue kind of setting, tissue construct which is implanted, and you are trying to look at the healing times or what exactly is going on, right? So preferably this is with binding and placebo as the control. Like I said, to brush up, control means that there is a control group of participants who receive no treatment, right? So there could be two set of groups, like one is not getting any treatment for a wound healing, and then you have a wound healing patch, which is in another group, and you do not know which one is which, like, right? Of course you have to know, but like the patients do not know what kind of treatment modalities are provided, and based on that you're assessing that how is the placebo group versus that treatment group getting affected, right? And there are cases that the placebo may perform better than the treatment group, and there have been reports where certain devices have completely failed. Or some treatment comparison studies, right? There could be some tissue engineered skin which has been placed here versus a skin graft, which is a traditional technique. You want to compare between those, right? Like these kind of things can be done. Randomized means that trial participants are randomly assigned to either of the treatment group or the control group, right? So that's important, irrespective of their demographics or what they are, right? Binding means that participants don't know which treatment they're receiving. This is single blinded, right? And some studies, neither of the doctor or the study coordinators, so that is double blinded. That is to make sure that there is no bias, right? So somebody has to know, that would be the principal investigator. That may involve a set of doctors and patients, right? So randomized controlled binding trials is the way to go for the most effective clinical trials. So clinical trials for medical devices, biomaterials, tissue constructs include control studies which has devices versus no device, right? This is an example. Similar tissue construct versus no tissue construct, or biomaterial versus some other standard material. Similarly, device versus gold standard device. Like I said, it could be a tissue constructed or a bioprinted skin graft versus a standard skin graft. Similar device versus gold standard device. Device versus standard care or physical therapy, right? So one case could be you just naturally allow a wound to heal versus you apply some kind of wound healing hydrogels to heal, right? So these are controlled studies. Blinding studies are usually difficult. The evaluator statistician can be blinded, right, to figure out that how the distribution is, right? Randomization can be done, right? And patient consent can be a roadblock in certain cases. The patient would like to know what kind of treatment modalities are being given. So this is, randomization is a challenge. So control studies and blinding is still fairly possible, but randomization, there are a lot of randomized control trials which are happening all around the globe, but randomization is a little challenging and daunting task for majority of the clinical tests. So types of devices for outcome selection includes therapeutic devices like clinical outcome, where you have more objective results than subjective results. Aesthetic devices, where clinical outcomes can be more subjective than objectives, right? And you can just replace the device terms with tissue constructs or biomaterials, like that would have the same norms. Again, diagnostic devices, which where you could have biosensors, right? Like for example, is focused on the diagnostic performance, which is objective specific, right? Bias and variance is something to be understood statistically, right? So bias is a systematic non-random error into the estimate of a treatment effect. So typically what that means from a philosophical level is, you know beforehand what the results are going to happen, or you know this is going to go in this direction. That means you're not doing the study properly or with preconceived notions. You have to eliminate, reduce, or estimate the bias so as to characterize in an accurate manner the safety and effectiveness of a device, tissue construct, or a biomaterial, right? So bias has to be clearly laid out. Is it a very intuitive bias that this particular, let's say, biomaterial or stem cell based treatment is not going to do anything? That could be a bias. Or it could be a bias, this is a 70% chance of doing better, let's try it out through the clinical trial, right? So the idea should be to eliminate, reduce, or estimate the bias, that what is the percentage or possibility, so that the accurate safety and effectiveness of that material or tissue construct or device which you're trying to use is properly estimated, right? Variance can be reduced by a more efficient design or a larger sample size. What is a variance? Variance is the outcome difference, right? So let's say I'm using this particular tissue replacement patch where I've used it on 20 uh, human subjects, right, 20 patients, and they have behaved in very different ways, right? 20% people, they are the healing is almost 3 times of traditional treatments. 30% people, the healing is worse than the traditional treatments, and 50% people, I'm looking at 5 times the healing, right, like of based on time of the traditional treatment. So that has a lot of variations, right? So variations should be reduced by having effective treatment designs or effective implementation designs on how things have to be done. So if I'm preparing a tissue construct, for example, it should be mechanically the same, bioprinting based the same kind of studies or assessments have to be carried out, and maybe some same sizes can be used for these kind of measurements in the human trial, rather than and maybe placing a skin replacement patch on the same location on the body to have a control location to get a similar set of results, or at least minimize the variance, right? If we are using very different environmental parameters, the variance could be very high, right? So I'm trying to plug in a wound with a new hydrogel based tissue engineering material. The wound is, let's say, extremely deep on the feet versus it is extremely shallow on the face. So if I'm going to treat both of them, they will give us outcomes which are very different, right? So something has to be controlled, that I'm going to only focus on the foot, I'm only going to focus on deep wounds on the foot, then you will get some similar results, right? So the bias and variance has to be reduced. Double masked or blinded randomized controlled multi-center clinical trials, or the RTCs, right? RCTs are the gold standard for clinical outcome studies, but not necessarily for diagnostic clinical performance studies, right? Such studies tend to minimize bias. Randomized controlled trials are pretty much good with the bias. They have low bias and variance. But these studies may be impractical, unethical, and unfeasible in certain situations. For example, there are some devices where it is impossible to mask anyone in the treatment, right? So there could be cases that you have a certain type of implant which can only, it's a patient specific implant or a patient specific tissue construct. Well, let's say the burn area, for example, is different. Coming back again to the skin grafting example. So I can very clearly identify as a doctor that I know this person with a 2 centimeter burn area is a different person. This is this XYZ named person, and this person with almost a 7 centimeter burn area is a very different person. So it is difficult to mask now. So this is very commonly occurring in randomized control trial. So it is very difficult to standardize these processes. So you'll have to look for millions of people, patients who have that same burn area. That's not possible. You're focused on the burn on an arm, let's say, that's the maximum you can focus on. The areas might be very different, and that might be challenging to do a randomized control trial in those cases, right? But nevertheless, like these have to be managed, and whatever the best you can do uh, to blind it and randomly do it. And majorly the idea here of randomization is not to include any bias or variance, right? That has to be minimized as much as possible. Blinding without masking leads to basically bias which needs to be addressed. Usually bias occurs if control is no treatment, right? So bias is highly likely. So if you're trying to just, if you have two or three different treatment modalities, it might be difficult to figure out the bias. But if you have a no treatment group versus a treatment group, you know as a fact intuitively that this is going to behave much more better, right? So that's where bias occurs. Bias is usually very difficult to estimate and can be quite large and extend over a longer period of time. So there could be less bias when you first administered a treatment. Let's say you did some grafting in 100 people, right? So the first 20 days, extremely no bias, everything is healing. But after 25th day, let's say 30% of the population is healed more than the others. Or after the 3 months duration, there is another set of population which has exceeded the expectations, and the other group has not exceeded anything. It is falling back, right? So then there is a huge bias there. When the study is non-doubtly masked, the influence of the patient or the investigator can be subconscious and unconscious. Sometimes you would know by back of your mind that this is how probably it's going to happen, right? So what happens when you do not have a randomized control system is you tend to take things for granted, right? That's where the study results get extremely skewed and do not show the real face of how it should look like, right? So these are some of the challenges if blinding is not followed. From a placebo standpoint, a placebo control is a totally ineffective treatment, but almost always masked for most trials. Placebo controls, sometimes known as shams, for devices can be sometimes be much more than difficult to devise than placebo pills, right? Placebo effect is well known for endpoints of pain or function and can be long lasting, right? So many a times you may have given some kind of a tissue construct or a medical device, right? Or you may have given something as a sham, right? It's a tissue construct which doesn't have the properties of the skin, for example. And then you have a biophilic tissue construct which is like skin. That could be a placebo and non-placebo kind of system, and you're trying to look at the healing effects, for example, right? So long-lasting effect with pain and function, these can be kind of characterized, and you can figure out besides the objective measure the subjective measures as well. Placebo effects are typically enhanced for devices, so medical devices typically have very distinct kind of outcome measures, right? And some of the placebo effects may completely enhance those effects, that without, in a completely untreated condition, it may show remarkably different results which are as expected, right? So that way you're not able to figure out the effectiveness of the treatment. Okay. Sources of placebo effect are typically from the expectation of a benefit, or sometimes with the showering of medical attention, which is known as the Hawthorne effect. It exists for objective as well as subjective endpoints and can be very long lasting. The sponsor should consider asking the subject and physician during the study which arm they think the subject is in, right? Like, are they showing like the right effect or the wrong effect? So sometimes these kind of things are asked like as a part of the placebo effect. There are one arm studies which depends on the objective performance criteria, right? Objective performance criteria, or the OPC, is a predefined criteria to check the effectiveness of the device or tissue construct or biomaterial. Usually a very well described and publicly available control is used to set the criteria, right? Like for example, heart valve studies, which are soft tissue mimics, like almost, right? Like in certain cases they could be rigid valves, but you're looking at heart valve studies, so there are some standard treatment modalities. What are the effects? They have been well characterized. So if you're coming out with a new design of heart valves, which has undergone some preclinical trials, and you're now looking at clinical trials, you have to have an objective performance criteria. It's very easy to do that because you have traditional data which is there to compare. But for exploratory studies, sometimes it might be difficult. So can be the upper bound or the lower bound of the confidence intervals. This is very similar to when we are trying to mimic the skin, for example. We have already ample of literature data on human skin where it has been mechanically tested. We know the upper bound, lower bound. So if we can find out a material which behaves within the upper and lower bound, we can call it a skin. Very similar to this one arm studies, right? Next is outcome measure. The choice for the most appropriate outcome measures depends on the primary objective, which is increase of benefit or reduction of harm specific, right? So state of development of the technology, feasibility, effectiveness, and efficiency are looked at. Quality criteria looks at validity and reliability. So if I'm doing a certain clinical test, is it valid? Validating a certain set of claims which I've obtained from my finite element studies or tissue characterization studies. Is it reliable? Is it something which is which I can predict for the 10th person, right? So that is the reliability aspect. So if something is not reliable, then it's very difficult to concretely say that. Acceptance by patients, physicians, and the scientific community. This is also important. There could be certain sensors and devices and tissue constructs which are built, and this is happening globally. Many of the bioprinted tissue constructs are not accepted by the surgeons, right? They say, oh, this is still in the research phase. We don't care unless this has been proven. Like you just keep doing it for research. So that acceptance might be missing, and that could cause a huge issue with the clinical trial or the acceptance of the probable material or the particular technology at a wider length. Then acknowledgement of the value of better tolerability or convenience. So there is this: is this particular material or tissue construct or device tolerable or convenience? So these are things have to be also looked at, right? It should not require a very drastic surgery. Objective performance criteria, if we define, is a numerical target value derived from historical data from clinical studies and or registries, which may be used for comparisons with safety and effective endpoints. So this is always a decision matrix. We need to always define our outcome measures by matrix, right? Like so objective performance criteria is very important for any sort of tissue constructs, biomaterials, medical device testing, because and surprisingly it's not there for majority of these categories because there is a lack of literature. So if you're trying to delve into a new industrial area, let's say we're all excited about bioprinting, for example, of liver, we should be really digging into what data is available. Has anybody been able to like develop a bioprinted liver which has not only tissue properties, which has the final set of outcome measures which is similar to an actual liver? If there are some properties, yes, we can possibly build that and compare with those properties, right? Let's say somebody has maybe bioprinted something in the US, like, and it has successfully shown in that clinical trial. I'm just giving in a random example. In that case, we can prepare that and go in that direction, right, for possible entrepreneurship and other events, right? And objective performance criteria can be carefully constructed from all available patient level data on a particular type of device or a construct or biomaterial. Elements to generate OPCs are identification of the medical devices or materials or the tissue constructs for development of the OPC, engagement of key stakeholders, right? Selection of data sources and performance of appropriate statistical analysis. Medical device market is very well grown in this aspect. So if you're looking at a classic hip implant, for example, there are a lot of data on what is a right hip implant, what are the possibilities, what can go wrong. These are very well designed, right? But if you're looking at a biosensor or something which is embedded within the human soft tissue or the body, that is where we have to be careful, because there may not be much of data out there, because this is the first time you're developing such a device. So what is the performance metrics which you follow? So this may not be known, right? So this is where we need to come up with an objective performance criteria. So in diagnostic clinical outcome studies, right, where we are trying to diagnose certain things, and we just spoke about biosensors, for example, this is used for diagnosis. Here the intervention is the outcome of the diagnostic test and the primary endpoints are clinical, right? It is usually always unmasked to the investigator but can be masked to the third party evaluator, right? If you're doing a clinical outcome study, the objective is to characterize the performance of the diagnostic based device on the test results from the subjects and to provide evidence to be used in the assessment of the benefit risk associated with the use of diagnostic device for the intended population. So for example, I'll give you a very classic example of smartwatches and biosensors, where it is uh, saying, basically it's claiming that it can measure the skin sweat, or it can measure the possible vibrations and other pulse readings on this underneath the skin, like in various regions. So this may not be able to do that. Or something is looking at basically the tissue degradation at certain point, let's say an osteoporotic biosensor, right? Is it able to actually sense that? Or tissue degeneration in certain areas on the body, could be soft tissue, is it able to look at that? So that's the diagnostic clinical outcome studies, and they have to be properly done. So what are the broad barriers in conducting clinical trials on medical device or constructs or biomaterials? Timing of assessment. In longer clinical trials of 2 to 3 months follow up, typically with the drug delivery devices, which are, by the way, also interacting with the tissues. So there is a lot of innovation in the drug delivery systems which has interactions with soft tissues. Usually a device becomes outdated by the time it trial is completed. So many new technologies are coming in, so by the time you have completed a trial in years, like new technologies have come in, right? This is quite common. Solution aggressive preclinical tests are required to reduce the follow-up period, right? So if you have an extremely solution focused aggressive preclinical trial, then this will reduce a lot of further steps into the clinical trial. What happens typically is everybody wants to jump into the clinical trial as quickly as possible with minimum efforts on the technology. That is where the lacuna is, right? We need to be spending majority of our time figuring out in a preclinical setting what is the best we can do and how the solution can be objective, and then only the clinical trial can be conducted so that the timeline is all reduced, right? Closely monitoring the post-market surveillance of such devices can be an alternative solution. So sometimes if you have reports or data on similar devices how or similar technologies how they have performed. So for example, if I'm looking at a skin replacement scaffold, I should be looking at how the skin replacement scaffolds have fared in the last 10 or 15 years. What are the data associated with? If the data are not very good, then I have to probably rethink what I'm looking at, right? Experience of the personnel carrying out the intervention and the learning curve of the personnel with the new device have to be looked at. So it's also based on the experience of the particular investigator, right? So if somebody without a knowledge of let's say soft tissue properties is looking at what are my outcome measures with this particular soft tissue construct, may not be able to find it out, right? Learning curve depends on the complexity of the device or the procedure or the material, right? Solution is reporting data in two phases, during the learning and after the learning. So this is the overall highlight. So during the learning, the data has to be kept on reporting, and that's one of the reasons if you look at our research group publishes a lot of papers in a certain area, like in skin grafting, for example. Whenever we have a new set of findings, we keep on publishing. That's how we are kind of completely active in that research for a while, and then we get the next stage that now we have done a lot of mechanical tests, so let's test it with other biocompatibility and other tests before we go for possible future clinical trial, right? So that's the way to go. Not that you publish one paper and that's the entire set of data and next step is some animal models and then clinical trial. It's a area of work which goes on for a while, right? Acceptability of the participants, reliability of the new devices or technologies, subjectivity becomes an issue when outcomes are patient or doctors reported. Blinding difficulties are other challenges which you will face often in conducting clinical trials, but they have to be taken with some caution, like while looking at what are the future outcome measures. Like in burn victims, blinding may not be an issue because you are aware of that the burn areas are different. Like could run a study on overall performance of the skin graft versus area specific differences in how the skin graft has performed over time, right? So these kind of things can be looked at. Choice of comparator, outcome assessments, these are several other norms and terminologies which can be looked at, right? I'm going to just skip over this, and there are several regulations and laws. Just to conclude, like central regulatory authorities and their formalities have to be completed while we are trying to run clinical trials. Stringent assessment protocols have to be followed before even the approval, right? Time duration between application and approval are sometimes a huge barrier to the clinical testing norms. Sometimes you may get an approval in a year and a half, right? Because your tests are not complete. It's just like a patent application, right? Like very similar analogous to that. So in those cases, you may run out of your resources to conduct a clinical trial at a later part because your funding has extinguished and things like that may happen. So there are several barriers there. So that kind of concludes our understanding on the different clinical testing norms. So now we are going to basically focus on some of the very interesting case studies, and we'll take some snapshots into something which we did and something which is done in literature into the soft tissues. We are just switching gears back after learning about the clinical testing norms. So this is one of the studies which have been recently done as a part of our research lab. So we wanted to basically look at skin grafting. So skin grafting typically deals with when you extract a part of healthy skin and kind of then run it through a meshing device, expand it, and then put it in a burn area site where the requirement of skin area is much larger than the healthy skin site, right? So this implantation typically requires a possible expansion in the healthy skin, right, which is not offered by the current traditional skin grafts, right? So you get less than 3 times expansion, less than 2.5 times ish expansion is the maximum which has been observed, right? So in order to gain further expansion, we started looking at different innovative designs or slit designs or cut perforation designs within the skin, which are known as metamaterial or auxetic based designs. So in order to look at that, we did a fresh cadaveric skin study. Why did we study fresh cadaveric skin? Because skin typically changes widely in properties as it dehydrates, like after death, right? So typically when a person dies, like after that, there is a certain time frame in which the skin gets extremely dehydrated, the body organs gets dehydrated, and the skin properties and every sort of properties keep changing, right? So typically if you store a skin from a cadaver who has died like 7 or 10 days before, or maybe 30 days before, you freezed it in a refrigerator, taken it out, tested it, the properties very likely are going to be extremely different from the real human skin properties. So that's why we have to focus on fresh cadaveric skin. So within 7 to 10 hours after death, we want to focus on extraction of that skin and testing it in the lab. So this is exactly the live pictures of this is a cadaveric skin. One of my student who is part of the PhD, like he has specifically been to AIIMS and in collaboration has been able to extract the skin samples and test with them to look at a skin grafting technology, which is the auxetic skin grafting, right? So these are some pointers here. So what we did was, this is just to give you a background. This is not to be taken as clear numbers. These are just to give you a background on how cadaveric tests are done. So ethical approval was obtained from the like the ethics committee. So this is some ethical numbers in 2023 from AIIMS Delhi to test the cadaveric skin samples, right? So this was the step one. So equivalent is you take the ethical approval from a hospital or from your institution, right? It has to have some medical focus in your institution to have an ethical committee or an ethics committee. Otherwise, you tie up with some hospitals or collaborators to get the ethical approval, right? So that committee typically looks at all the aspects of your study design, how many samples, how many cadavers, how many skin samples you're going to test, what exactly is the amount you're going to extract, what is the size of the skin, and things like that, how you're going to do the extraction, transportation, storage. All these things were looked upon, right? So after this, once this proposal was through, we could uh, take out fresh cadaveric skin from the Department of Trauma Sciences in AIIMS, like where typically we had a collaboration, and they basically, here you see a lot of trauma cases where it is a person is dead on arrival, and basically within the 7 or 10 hours of it, you get fresh cadavers which are with all the help from AIIMS, like we were able to collect this, right? This is from the uh, from his one of his work. It was ensured that the skin collection and mechanical testing was conducted within 24 hours of the death. So that was the general norm, because it's difficult to procure cadavers which are just within the 9 or 10 hours. So 24 hours was the overall consideration, but we still maintained like below 12 hours as much as possible. So we could remove 20 samples of skin from the thigh region. So that's the control. We are trying to only focus on the human thigh region here, just to make sure, because skin is widely different. Remember, like we have talked about skin at length. If you take a piece of facial skin versus somewhere else, it is anisotropically different, thickness is different, so the material properties are widely different. So we wanted to focus on the human thigh only, and we took out 20 samples from the thigh region of the human body from 7 cadavers. So we could only get 7 fresh cadavers, because we could also get more, but that was the study statistically, that we wanted to at least test on 20 samples from 7 cadavers, right? We had to specifically look at the average weight, height, and age of the cadavers. So 69 kilograms was the average weight, 6 feet was the height, and 47 years was the approximate age, average age, right? In the thigh region, we also, before we take the skin from the cadaver, as a part of the ethical approval, we have to make sure that whenever a new fresh cadaver we are going to use it for skin extraction, we have to take informed consent. Now, how do we take an informed consent from a cadaver? You have to take an informed consent from somebody immediate to their family. If somebody has basically declared that the person who has died is part of their family or family member, that is number one. Otherwise, in case of undeclared bodies, that is when you get a consent from the doctors typically. So there are standard practices and rules which have to be followed to get a proper consent for the cadavers as well. So if you're going to ever run a test on human cadavers for soft tissue testing, these are some of the standard things which you will find, right? So this is just to give you that overall idea. This is, by the way, a much more longer study than this, but these are some initial findings which we came out with, right? Okay, so what we did next was preparation of the skin samples. So total 20 samples of the thigh portion of 7 fresh cadavers, as we said, was collected. Written informed consents were obtained to perform mechanical testing of fresh cadaveric skin, right? The next control was we made the size of the skin sample which we extracted similar. So it is difficult to keep it similar while extraction, but after the extraction we cut it out, we trimmed the edges to make it all square, right? And kept the same size, which is 60 millimeters or 6 centimeters by 6 centimeters by approximately 1 millimeters, which 1 millimeter is focused on. This is basically a blade. It's a cutting device which is used for skin extraction, that was set with a blade length of approximately 1 millimeters, which is going to extract in a split thickness way the entire epidermis and a very minor part of the dermis, because epidermis is usually around 0.1 or 0.2 millimeters, right? And then you are looking at a dermis of usually 1 to 3 millimeters. So it's partial dermis and total epidermis. So this is what you're looking at, right? So this was excised using a surgical instrument, which is a standard surgical instrument for excisions. Excision is taking out or extraction of skin. The samples are as shown here, 1 to 20, right? So the cadaveric skin, thigh region, age, weight, and height used for the studies are mentioned here. So sample collection was done. The next step was preparation and testing setup for skin graft samples. So what did we do next? So we had used this innovative method for development of traditional and auxetic skin grafts, right? So a 3D printed mold. So we use a 3D printer to design and print a mold was developed to create skin graft patterns. So this is the skin graft patterns. So you can look at the graft preparation pictures on the left side bottom. So there is a traditional pattern and there is an auxetic pattern, and we had basically printed this and we got molds with this cuts, right? And then what we did was experimental study was performed on the traditional skin graft simulants, right? So the molds acting as templates and surgical blade was used to completely perforate at these locations of the cuts the skin, and based on that we got bunch of slits which are properly cut, right? This is what you will see for a skin graft which has come out of a meshing device also. Typically you would see a skin sample which has come out of a meshing device having all these cuts. That is typically translated from a meshing roller which has these perforations or cut blades, but in this case we just cut it with a surgical blade, right? So after doing the surgical blade cutting, we could create a traditional graft which is the traditional slitting pattern, and an auxetic graft which is an auxetic slitting pattern. So the traditional and auxetic graft patterns are shown on the figure on the left bottom. Then we did some testing, so uniaxial testing on a universal testing machine, right? So this is a testing setup where these graphs are getting placed. We also did a biaxial testing because skin usually has the effect of both uniaxial and biaxial forces, like based on. So if you have like a completely symmetric wound, you will have more uniaxial forces. If you have a completely unsymmetric wounds, like you will, sorry, it's the opposite. If you have a completely symmetric, you will have biaxial forces. If you have a unsymmetric wound, you will have more of uniaxial forces because you have a longer length of the wound at the other direction, right? So that is typically what is observed. So there is uniaxial and biaxial tests which are conducted using two devices. One was a standard UTM device, and the second one was a custom built biaxial device, which I might have explained in one of the lectures, right, for biaxial testing. These are some of the results. So what we did was we wanted to compare using a skin simulant material, right? So we tested the skin from the thigh portion. This is the plot number 1 on the top, right? Skin from, and this is a Makode et al., like where the original skin from the human skin properties were tested. Skin from the thigh portion was tested, and then a skin simulant material which we had developed earlier in the different lectures we have shown, that was also tested, and we could see some similarities. We have to make the displacement rate or the strain rate constant here, and we looked at this stress strain profile, and these are the test results, like with error bars, because every test has been conducted at least with this multiple samples, right? So skin simulant is the lowest property, followed by the skin thigh portion and the skin Makode et al., right? Next is B, is when we are looking at traditional skin graft properties and auxetic skin graft properties at the same strain rate of 24 millimeters per minute, which is fairly at the lower end. It's almost a quasi-static kind of test, right? Low strain rate testing, it's very slow testing of tensile test, right? So we could see some difference in the properties on the traditional skin graft and the auxetic skin graft overall. However, when we looked at the skin properties at a higher strain rate and just the skin, so the left plots, like the middle plots which you're looking at, is just the skin, and the right side plots are with the graft, right? So that is with skin with the slit, right? So you're just looking at the second plot in the middle, the bottom plot. You're looking at skin Aniyad et al., which is at a high strain rate they had tested, and skin experimental, which we have derived from the cadaver. We are seeing very similar responses, right? Like with the R square of 0.81. So they are quite similar in terms of stress strain profile, which indicates that the skin which we have derived here is actually from a fresh cadaver and is similar to a live human skin. This is what this plot tells us. Next is we look at traditional skin grafts and auxetic skin grafts. When we look at at a high displacement rate, at a low strain rate, what we have seen is there is a slight difference, but at a high strain rate, there's a huge difference between traditional skin graft and the auxetic skin graft. What we do observe here is the stresses in a traditional skin graft is much more higher, significantly higher than the auxetic skin grafts, which is a positive. In the auxetic skin grafts, the lower the stresses, the better it is in terms of expansion. And what are the key results from the biaxial and meshing ratio? In the E part, we are looking at traditional skin grafts and auxetic skin grafts at two different displacement rates, right? So what we did see was at 24 millimeters per minute, the auxetic skin graft was behaving much more with a higher meshing ratio, expansion ratio than the traditional skin graft. Same was observed with a higher strain rate as well. So irrespective of the strain rate, what we did see was the auxetic skin grafts were able to expand much more greaterly compared to the traditional skin grafts, which itself is a groundbreaking news for the community, that auxetic skin grafts do have the possibility of having a better expansion. By the way, like this research on just some preliminary data which we have collected on the cadaveric skin was conducted after we have almost published like 10 plus articles where we have looked at the different designs of the the Oxatex. We have compared it with traditional graphs. We have done finite element studies. We have done lots of experimental studies with skin simulants. So after all those study validations, once the research idea or hypothesis was very clear that the auxetic uh, perforations can actually lead to higher expansion, and in few cases we did not get that. We have to optimize and find out the right auxetic properties or shapes which lead to that higher performance or higher expansions, right? Once we had done all that research, then only we came to this later stage of preclinical work where we have looked at cadaveric skin, which needed all sorts of clinical testing attributes, which we have so far learned in this lecture and the previous lecture. So all those norms were followed, as well as the ethical norms were followed in order to get the preliminary data with cadaveric skin, right? So these are some of the findings, and we could finally validate that auxetic skin grafts do work better than standard skin grafts. So now maybe with further tests, this is a very preliminary study. With further tests, further samples of testing, if we can test skin from different locations of the body and can keep on validating and have a similar result, then we can potentially move into some further preclinical trial into animal models, and then possibly into some clinical trials with human models in the future. So this is one example of the implication of clinical testing norms and ethical norms into the soft tissue testing, which we have implemented in our real life scenario. I'll quickly take a snapshot of a few more one-page case studies just to kind of showcase some of the varieties of soft tissue testing clinically, which needs to be done like as per the norms, right? So this is another example. This is from an article, load deformation characteristics of aceticular human scalp, assessing of tissue grafts from a material testing perspective. This is a article in Nature, I believe, which is one of the very reputed journals. So here basically a tissue from an aceticular human scalp is the top part of the head. From that, a skin sample was retrieved. So after the retrieval, there was shaving which was done to remove that hair, shaping which was done to cut it in a particular shape, like in a dog bone kind of specimen shape, which is a common norm for doing uniaxial tensile test. Then finally, cell removal was conducted to just focus on the tissue aspects which will depict the right properties of the material. Then uniaxial testing norms were followed, and a lot of tests were conducted to look at the fracture. So overall, the uniaxial tensile tests were conducted until failure. So this is an example where soft tissue testing was conducted in a with cadaveric tissues, right? Like where all the clinical testing norms are followed. So you welcome to read on this article or very similar articles to read about the clinical testing norms with cadaveric tissues. This is another example which is from this paper on the tear resistance of skin. So this paper is focused on studying cadaveric skin and basically how they tear or how they rupture. So you can look at ABCD, the top figures, which looks at how the tear propagates as there is a uniaxial tension applied to the skin, and using this they have been able to simulate some similar material properties and find out important attributes. This is again a test, more of a preclinical test, which is based on cadaveric or soft tissue experiments. This is another example where this research is titled as characterization of main pulmonary artery, right? So this is from an organ, and valve annulus region of piglets. So this is again an animal model which we have often seen in preclinical tests using echocardiography, uniaxial tensile testing, and a novel non-destructive technique, right? So what happens here is we are looking at taking out samples from this particular location, pulmonary artery, and testing them on a very specific uniaxial testing measurement device, which has been built specifically for dealing with these kind of softness tissues, right? So you cannot have a huge machine deal with these kind of tissues. So you can see very specified clamps which have been developed along as an attachment for this tissue. So this is another example of animal models which follows all the clinical testing norms and ethical norms. So you can also read up this paper to look at the clinical and ethical testing norms, right? This is the last one. I wanted to showcase that cadaveric and animal model tests are not only conducted for healthy tissues but also for diseased tissues. So this is where they are looking at uh, uh, biomechanics of mitral valve leaflets, second harmonic generation microscopy. They are looking at biaxial tests. So this mitral valves were extracted, and biaxial tests based on the actual loadings which are applied to these kind of tissues, like in a realistic setting, were kind of modeled through like mechanical tests on this realistic tissues, like following all the clinical norms. And this is of the diseased tissue, characterizing the viscoelastic properties of human melanoma, which is a type of skin cancer tissue, using Croney series. So here you're looking at a tumor or a component of cancerous skin has been extracted, and they are doing some indentation and compressive tests on this physical form using all the clinical testing norms which we have learned so far, and trying to figure out how the diseased tissue is performing as compared to the original skin. So there are a lot of results covering this. So long story short, as a part of the conclusion, what I would like to say is cadaveric tests, any kind of animal model tests, and human based tests have to follow all the clinical norms, clinical testing norms. And it is not only the mechanical tests which are conducted, a lot of other things have to be followed. And as far as the mechanical tests are conducted, there are certain protocols to be followed for storage, for transport, for testing, to maintain the safety and adherence of the protocols as per the real tissue norms, right? So with this, I would like to conclude, the testing of soft tissues from a clinical standpoint. So thank you.